Okay, this week you're going to be doing a bacterial transformation. Recall in class we were talking about the different types of genetic recombination that can occur naturally with bacteria cells, but also can be done in a laboratory. We talked about transduction, we talked about conjugation and transformation. So you're going to be doing transformation. Remember, conjugation was with the use of a sex pilus. If a bacteria has the gene to produce a sex pilus, it can actually transfer its plasmid to a different bacteria. Then we had the transduction, which was the use of a virus where a phage virus would come in and it would pick up some of the DNA from one bacteria and transfer it to another bacteria. The transformation that we're going to be doing today is similar to the one where we were looking at the bacteria that was pathogenic and the bacteria that was not pathogenic, and heat killing the bacteria that was pathogenic, and the DNA from that bacteria was released into the solution, and then the non-pathogenic bacteria picked up that gene and was able to express that pathogenicity, maybe a capsule or something like that. So what we're going to be doing is we're going to be taking E. coli. E. coli in itself is non-pathogenic. It's something that lives in your gut. There are strains of it that are pathogenic, but normal E. coli is not. Normal E. coli is also not resistant to antibiotics. So as part of our transformation, we're going to be giving it some special abilities. First of all, we're going to give it the gene for ampicillin resistance. Ampicillin is an antibiotic. We're also going to be giving it a gene called the LAC-Z gene, which is the gene for the beta-galactosidase enzyme, which uses the sugar in the media and will cause a blue precipitate, so you'll have blue colonies. Okay? This is similar to the blue-white cloning lab that we talked about in lecture, except for we're not actually inserting that gene that interrupts the LAC-Z gene. So we're only really doing the blue part of it, but you might see some white colonies expressed also. So for this experiment, it's going to be not extremely time consuming, but there's lots and lots of steps, and there's a wait period of 15 minutes here and there and things like that. So it's not a quick run through lab like some of the ones that we've done. There's going to be lots of different supplies. This is the majority of the kind of supplies that you're going to use, but not necessarily the number of everything that you're going to use. Okay, so first of all, let's start off with what we're going to be doing first. So the first thing that you're going to do is you have two sterile tubes. These two tubes that we've had cultures in, but these are sterile, they have nothing in them. You're going to label them DNA positive and DNA negative. Now the DNA positive and negative is stands for the fact of whether or not you're going to put the plasmid in. The plasmid, you remember, is a circular DNA that's different, not part of the normal chromosome of the bacteria, and it's going to carry particular genes. This is a plasmid that is manufactured to have the ampicillin resistance gene and the LAC-Z gene, but also has some different sites that you can put a restriction enzyme cut in, okay? But since we're not adding that gene, that's not quite as important, but definitely look at the book at what that plasmid looks like, okay? So then with these two particular tubes, we're going to aliquot 250 microliters of our particular solution into these tubes. Now the solution that we're using is called calcium chloride, and it needs to be kept cold. So when you come to your table or when you get your stuff from your station, the calcium chloride will actually be on ice. You'll have much more ice than this, but I was just trying to keep things minimal for today. It will be labeled CaCl2. It's going to be in one of these little smaller tubes, even though we're putting it into a bigger tube. You're going to need to aliquot 250 microliters. For this particular one, you're going to use one of these transfer pipettes, but these are sterile. So make sure you open it from the bulb end rather than the tip, okay? And on these particular pipettes, it's a 1 ml pipette. So the very top mark is 1 ml, the middle mark is 0.5, between the 0.5 and the bottom is 0.25, which is 250 microliters. So you're going to that first notch, not the very end of where it gets fat, but the next notch up. You can see this one has a 5 and that one has a 1. We're doing half of 5, so we're doing 250 microliters or 0.25 mils. okay? And once again, we want to be as sterile as possible. When using this pipette, you squeeze it before you put it into the solution, and then as you slowly bring it up, you go to where you need to go, and you put the same stuff in both of your tubes. We're not flaming these tubes right now like we've done in the past. Okay, I'm going to get another 250. And probably don't need to do the sterile technique we've done in the past. Well, we do need to use sterile technique, but we could probably take the lid off without lifting it. Okay, so that's my 250 microliters of my calcium chloride. Okay, we're not going to use this pipette anymore, so we would throw it in the trash. It doesn't have any bacteria on it, so there's no need to put it in any type of biohazard container. So we're putting the exact same stuff in both of these tubes. The next thing that you're going to add is going to be the E. coli. So for this particular procedure, we're going to need our flame. It says you can use plastic sterile loops, but we're going to use our regular loops that we always use. Okay, and we need to flame it and use our sterile technique that we always use. Okay, we're going to let that cool off. Now what it tells you to do is you'll have a plate like this that has E. coli out on it, and this is going to be your starter plate. Okay, this plate will have separate colonies of E. coli, hopefully. If they don't get out in separate colonies, we'll just do the best we can. They use that streak plate method. We're going to want to take one very large colony, about 5 millimeters. So if you can think what a 7 meter is, it's a half. It's a half of that centimeter. Okay, letting that cool off. We're going to take a colony. Like I said, a good sized colony, and you're going to put it in the first tube. You want to actually break it off in there. Okay, you want it to all come off. Okay, we'll flame that. And then we're going to do the same thing. You want to get basically the same amount. These two tubes need to be identical, except for adding the DNA. Another good sized colony. Now remember, don't gouge the auger. If you gouge the auger, you're going to end up with auger in here, and it will mess up the results. So you want to break it off on there. Make sure it comes off. And you want to have a milky solution. So I don't know if you can tell, but there's definitely a lot of bacteria in there. If it still looks clear, you haven't added enough, so you'll need to put more. So I put the calcium chloride in both tubes. I put the E. coli in both tubes. Now at this point, I don't need this right now, so I'm going to turn that off. Okay. Now we're going to pipette 10 microliters of our plasmid. Now the plasmid will probably also be on ice in the same container that the calcium chloride was in. Now we're going to use these micropipetters. Our pipetters go from 2 to 20 microliters. The 2 is the lowest that can go, and 20 is the highest it can go. Never go past 20, so don't go to 21. Never go below 2, don't go to 1.9. On this, you'll see there's a window, and there's 2 black numbers and a red number. Okay, the black numbers represent the numbers above the decimal point, in front of the decimal point. The red number represents a number after the decimal point. So the black numbers, so for you to have 20 microliters, it would be 2, and then there would be a red 0. That's 20 microliters, so that's a decimal point there. We want to have 10 microliters, so we're going to set our pipette at 1, 0, 0. Okay. Now, like I said, don't ever go above 20 or below 2. So I'm at 13, so I'm going to go up. No, I'm going to go down. 10 is less than 13, isn't it? Okay. So now I'm at 10. It says 1, 0, 0. So make sure that's set the right way. You could even set it up before you get started. So we're going to aliquot 10 microliters of our plasmid, which will be in another microcentrifuge tube, labeled plasmid or P or something. I'm not sure how it'll be labeled. Okay. 10 microliters is a very, very small quantity, so we don't use a regular pipette. And when you use the pipetter, there's two stops. I can press down and I feel resistance, but then I can press further. When you go to pull up the liquid, you go to where you hit your first resistance, put it in the tube, and slowly let it come up. And you can't see what's in here, but it is only that amount, the very tip of the tip. It is a very small quantity. So now we're going to put that into only the tube that says DNA positive. Now you want to make sure you get it down in there. Then we're going to put this in a waste container because now there's possibility of it having bacteria on it. You do not put the plasmid in the one that says DNA negative. The other thing that you want to do is make sure you tap those and make sure everything gets mixed. Okay, if you have stuff up on the sides, it's not going to react. Okay, now you're going to take and you're putting these guys on ice for 15 minutes. I'm not going to wait 15 minutes. However, I'm going to stop the video now and then I'm going to start again because it might end up being too long and I won't be able to post the whole thing.